Good morning, everyone. Bonjour à tous. I'm the Consul General of France here in San Francisco, and I'm very honored to welcome you to the opening of this second edition of a science fair on healthcare. This science fair is co-organized by UCSF's Quantitative Biosciences Institute, QBI, and the scientific team of the Consul General of France in San Francisco. I want to thank this scientific team and the teams of QBI for their dedication and hard work in organizing this science fair. And I'm grateful to all the panelists who will kindly offer their time to share with our viewers their sharp insights to make this event a success. We will have a chance to hear from doctors, researchers, and entrepreneurs, and they will show how crossing borders, disciplines, can provide synergies to tackle future challenges of healthcare, such as immunology and parasitology, among others. Computer sciences, data, art, mathematics, and healthcare are disciplines that matter a lot to our two countries, France and the US, and in which, on the eve of rapid and promising developments, we have mutual interest to strengthen our collaborations. Such cooperations can only be built on mutual understanding of trust between individuals over the long run. So just like last year, I hope that this week's event will allow to open a series of meetings and exchanges which will allow new links to be formed. And I have to mention in this context that, as you might have heard, there could be a stronger collaboration in the months to come between the Institut Pasteur and QBI, with a standing Pasteur presence at QBI. This is great news, and I think it will increase the strength of both institutes. The synergies between them are very clear. QBI provides outstanding quantitative and innovative technology, and the Institut Pasteur provides a cutting-edge expertise on infectious agents, and this is known around the world. And this reinforced bond, which would also be the logical continuation of an extremely fruitful cooperation, is made which made it possible to perform remarkable therapeutic repositioning against COVID-19 in the past months. And I can't stress enough how tremendous it has this collaborative work has been, which took place in the past months, when it should actually have taken years in normal times. So this initiative between Pasteur and QBI is in perfect line with the motto of this week's science fair, which is crossing borders and disciplines for healthcare. Allow me also to take the opportunity today to talk about the upcoming visit to San Francisco of a French scientist, entrepreneur, and ambassador, Raphaël Torgman, in January during the JP Morgan Healthcare Conference. She is the Director General of a biotech investment fund, and she has been tasked by President Macron to promote the French biotech ecosystem. And I would like to stress that there are a lot of exciting projects and opportunities in France, and I encourage everybody to meet Raphaël Torgman during her stay in the Bay Area. Finally, I would like to pay tribute to each of those at UCSF and elsewhere, everywhere, who are committed to improving the quality of care through innovation and research. Thank you all for your attention, and enjoy this week's program.
Thank you, Frédéric. Bonjour, good morning, everyone. It is my honor to be opening the second annual science fair hosted by QBI in the French Consulate of San Francisco. We actually started working with the French Consulate back in 2019 when we had our very first joint symposium with the Institut Pasteur and invited them to be a part of it. An additional fantastic outcome of the relationships that we have formed with our colleagues in France has been the flourishing and very progressive collaboration we have had with the French Consulate right here in San Francisco. At QBI, we pride ourselves in breaking down silos and building bridges across those silos. And really, this particular bridge between our cultures has been incredibly fruitful for a number of different impactful discoveries. Together with our partners at the consulate, we've been hosting a number of events focused on this international interconnection in the past 2 years. I am proud to say that the pandemic not only did not stop us, it actually made us a more collaborative and creative force. At the onset of the pandemic here at QBI, we formed the QBI Coronavirus Research Group, QCRG, composed of hundreds of different scientists at UCSF to try to find answers to the SARS-CoV-2 pandemic. One important issue at that time is that we did not have a BSL-3, a biohazard safety level 3 facility where you could actually grow the virus available to us in the Bay Area. But the Institut Pasteur not only had one, but they also had some of the top virologists in the world. And this is what made this such a great collaboration. And this is what Frédéric touched upon in his opening remarks. So the consulate played a key role in assisting us in getting our compounds and drugs to Paris so that we could test them to see if they have any antiviral effects, despite embargoes and travel lockdowns. And really, thanks to this very key collaboration, we were able to make incredible and fast progress. This is but one example of what we know that these collaborations can foster. This science fair, which we co-host, is a continued attempt at building creative bridges with academia, medicine, and industry across borders. We are excited to continue this new tradition, and we hope that you will enjoy a series of robust panels this year. Thank you for your attention, and please enjoy.
Thank you very much, Nevan. Thank you, Frédéric. Hello, everyone. I'm Jean-Baptiste Bordes. I'm the Attaché for Science and Technology in the French Consulate General at San Francisco. Welcome also to the panelists. Thanks to Aude and Julian to take their time to share their expertise. As today we will talk about immunotherapy. So immunotherapy is becoming a more and more mature set of approaches for curing a wide range of ailments, from cancers to autoimmune disease. It's usually based on a highly personalized treatment for specific markers and targets. And in this panel, those specialists will explain how its rise comes from the successful and powerful combination of several disciplines: immunology, statistical analysis, and computer science, just to name a few. So first, I will let Aude introduce herself.
Hello, everyone. My name is Aude Chapuis. I'm actually a Swiss person. I trained in Switzerland, in Lausanne, in med school. And I moved to the United States now 18 years ago to essentially do a postdoc in research in immunology. At that time, I was actually on a project for HIV immunology because that was the big thing at the time. And I switched in the United States to study T-cell therapy for cancer. And since 2003, I am at the Hutch in Seattle, which is a big research institute. And my research focuses on T-cell therapy using T-cell receptor therapy as opposed to CAR T-cell therapy for multiple cancers. And we have translational programs spanning viruses, essentially targeting, for example, the Merkel cell polyoma virus. We have also targeting overexpressed self-proteins in AML and in solid tumors, such as Wilms tumor antigen 1, as well as other cancer testis antigens that we are targeting. So essentially, my lab really focuses on the translational potential and seeing from the difficulties that we had, for example, of cell persistence in the humans. We actually had to go back a little bit to the lab. So really, this bench to bedside and back scenario in order for us to really improve both the targeting and how we target these cancers in patients. So that's my research.
Thank you very much, Aude. Your turn, Julian.
Well, thank you, Jean-Baptiste, and good afternoon, good morning, everyone. It's a pleasure to be here. My name is Julian Mahmet. By way of background, PhD in molecular biology and genetics in France. I did my PhD in France at l'IPMC at Nice Sophia Antipolis. And then kind of just like Aude in the early 2000s, actually moved to the US for postdoctoral training at a place called GNF, the Genomics Foundation, Novartis Genomic Foundations, where I did my postdoc training. And then I was in San Diego, relocated to San Francisco to work for venture-backed companies in the last 10 years. So in terms of expertise, about 20 years of expertise in drug development, really from like early stage drug discovery, concept development, all the way to mid-stage clinical trials. In terms of areas, therapeutic areas of interest, that span from pain, neuroinflammation, neurodegeneration, inflammatory disease, and oncology. When it comes to oncology, I have a long-term interest in understanding the complex network that kind of make a cancer cell what it is and make it grow and proliferate in the body, and trying to understand the structure of those molecular networks, their hierarchy, and how we can treat them for broad effect from a therapeutic standpoint. So happy to be here today.
Thank you very much. So now let's talk a bit more about the science. We know that immunotherapy is working better and better, and actually when it works extremely good. However, there are still a set of limitations. So in your opinion, what are the limits of today's immunotherapy? What are the next frontiers in terms of research and developments?
So maybe I'll take that question.
Yeah.
So, obviously immunotherapy has taken a huge leap in advance, first of all, because of the checkpoint inhibitors and anti-PD-1, anti-CTLA-4, and now there's a whole bunch that are being developed, which really sort of use the immune system of the patient that is already present to try to boost that and try to attack the cancer. Then the second really big leap was with the chimeric antigen T cell targeting CD19, which is a marker which is expressed on B cells, all B cells, including tumor cells, lymphomas, and leukemias. And that really was a huge leap. I think at that time, when the first papers came out in 2013, everybody realized, wow, the immune system really can have an effect. And so a lot of effort has been going into trying to find other targets, especially for solid tumors. Solid tumors is really I would say a great barrier because they have immunosuppressive tumor microenvironment. And in the end, there aren't that many targets that are expressed on the tumor cell, but not on other normal cells. And so now, a lot of research is going into trying to find, for example, CAR targets, chimeric antigen targets for solid tumors. But there hasn't been a huge breakthrough at this time. Whereas, for liquid tumors, ALL, and also myeloma with the BCMA CAR T cells, really there's been a great expansion. 80% responses. I mean, it's quite remarkable. So I think right now the field is switching a little bit, and more we see trials that target HLA peptide combination. So basically, with the physiological mechanism with the T cell receptor. And this has the advantage that essentially you can target any surface or intracellular protein as long as it is presented on the surface of the tumor cell. And so there you can start targeting mutations specific to the tumor. You can start targeting overexpressed self-antigens, cancer testis antigens, which, for example, are primitive genes re-expressed in tumor cells where they're not expressed on normal tissues. This modality, I will say, also has its limitation. It was actually present before the CAR T cell, and there wasn't a lot of funding for it because it didn't work very well. But now there's a renewed interest, and I think that there's a lot of effort across Europe and across the United States to try to make that type of therapy work. The disadvantages of this type of therapy, I will say, is that you're limited by the HLA because we have many HLAs on our cells. We're a very heterogeneous population, and every T cell receptor will recognize a specific T cell HLA peptide combination, and so you have to choose the right one. The other aspect of T cell therapy, I will say, is that contrary to the CAR T cell, what has, first of all, it signals in the cell, and then it also has what we call a signal 2, which essentially boosts the cell. And in normal cell, this signal is activated when, for example, CD28 binds to CD80 on a tumor cell or an antigen presenting cell, except that this signal is not present in many tumors. And when you do TCR therapy, you're only just plugging in the key, the very specific key, but you're not giving it the additional boost. And so now there's a lot of effort to try to not only try to tether a signal to the TCR target to try to boost this type of therapy, and also, for example, engaging other cells of the immune system, such as helper T cells, in order to try to make the cells, not only persist longer, but essentially be there and be able to function for long periods of time. So these are the current challenges in T cell therapy and CAR T cells, I would say in 2 words. But we're getting there. I think, I'm not sure we're going to be there in the next 5 years, but definitely we're making huge strides towards making these therapies potentially therapeutic down the line.
Thank you. Julien?
So maybe to build on Jean-Baptiste and introduction and Aude's feedback, just from a macroscopic standpoint, so immunotherapies have been here now since 10 years. I think the first one was approved probably in 2011 for the treatment of melanoma. Since has been great progress as a class. There have been like 6 compounds approved and probably now 15 to 20 indications are being covered. So tremendous effect in some areas where there was a lot of resistance to treatment. That's kind of the key benefit for immunotherapies. Now for the limitations, there is, as kind of Aude was saying, kind of a breadth of applicability issues. There are still to date, I think, no more than 40-45% of patients with cancer are eligible for this kind of therapy. So that's one key limitations that come from kind of 2 angles. One is they're being developed indication by indication and subtype of cancer by subtype of cancer. So it takes time to bring more patients being eligible for this. There's also the problem of the toxicity of those drugs. While they work very well, they can be very toxic. That goes from minor toxicity, but all the way to catastrophic immune responses that leads to death. So it's a wide spectrum of toxicity. And for some cancers, that's very difficult because if you take, for example, liver cancer, patients are not healthy in the first place. It's not like a cancer like breast cancer where the patient is healthy. It's a healthy tissue that becomes a cancer. In the case of liver, it's 10, 15 years of hepatitis and inflammation that leads to a cancer. So the patient is pretty bad shape to start with. So the tolerability level is way low. So the toxicity of those drugs is another one. And the last one is the viability of the responder rates. We still are, and that's one of the big efforts being done today with precision medicine, but the responder rate goes from anywhere from 5 to 10% of patients all the way to 60-70% of patients, depending on indications. And we don't have a very good handle on this. So I think that progress on these 3 fronts, like precision medicine, to understand better, anticipate better, the patient response is kind of key in this area, and also finding combination therapy that would allow lower doses and increase the tolerability. I think those are the at least two key axes of innovation in the space.
Thank you. And how can we build some compounds that can attack several targets at the same time with immunotherapy? Can you elaborate on this?
So right, maybe, do you want to start on this? I'm happy to start.
Yeah, well, so I mean, for example, our compounds really are sort of either chimeric antigen receptors. So basically, you have an antibody linked to the intracellular portion of the T cell receptor with CD3, CD28, or 4-1BB intracellularly. And so I think there's sort of 2 avenues of research, right? One is, can we find better antibodies that, in terms of what to target? The CAR T cells are obviously bound to recognize something which is expressed on the surface. So there's a huge effort, I will say, to try to find new targets or even targets that are, if one's expressed but not the other, then the molecule will not trigger. And so there's really a very large breadth of research in this area. In the TCR world, it's a little bit different. So the challenges there, I will say, well, first of all, you're limited, as I said before, by the HLA, right? So so you have to find right HLA and peptide pairs that you want to target. The second part is a lot of tumors actually downregulate HLA in order to escape the immune system. And we think that potentially the checkpoint inhibitors can actually make that worse. And obviously, these type of therapies, most of these trials are done in patients who are resistant to checkpoint inhibitors. So a lot of effort also there to try to upregulate. Most of this is epigenetic, meaning that it's not, the HLA loss is not embedded in the DNA. It's really at the transcriptional level. And so a lot of effort there going to try to upregulate this HLA. And I think that's going to be key in order to make it work. Now, in terms of trying to find, what are the molecules that we're going to be using, there's been a lot of effort to try to find what is the best way to find a T cell receptor. Some effort has been going into, for example, raising or identifying a T cell receptor in a mouse. So basically, you transduce. It's an HLA transgenic mice with the HLA that you want, and you express the protein that you want to target. Except that a lot of these proteins are actually not expressed in the thymus of the mouse. So basically, the T cell receptor that you generate has a very, very high affinity for that target, and could potentially also be what we call promiscuous, meaning that it can now not only see the peptide HLA combination that you want, but also different ones. And that has been a cause for toxicity a few years ago at the NIH, where they targeted MAGE-A3, which is a cancer testis antigen, and unfortunately, also there were brain toxicities in this type of approach. Another approach is to try to take a T cell receptor that recognizes a peptide HLA and then start, playing around with its sequence, and to try to affinity enhance it. So basically, trying to make it more sticky to the HLA peptide combination. And that also has been done, except that there was also toxicity. And so I feel that this is sort of an avenue that is sort of being a little bit abandoned at this time, because, while you do that, you can not only modify the piece of the T cell receptor that recognizes the peptide, which is what you want a higher affinity towards the peptide, but also starts to recognize different HLAs, and that can cause off-target toxicity. And indeed, that's also what happened at UPenn with heart toxicity. So a third approach, and I think that's what people now are more looking for, are looking for these T cell receptors in patients who have recovered or have had some control over their tumors, with the idea that if the patient has been able to control his tumor, ultimately the T cell receptor, let's try to find that T cell receptor that mediated that, or to try to find these T cell receptors in normal donors, matched donors, HLA matched donors, and find sort of as a needle in the haystack to try to really understand and find these T cell receptors. It's just numbers game. If you take 15, 20 donors, you might find a T cell receptor which has the high affinity. So I think that's where the research right now is going in terms of finding the molecules that ultimately will be brought to the clinic.
Julien, your point of view?
I fully agree with Rod. Maybe just to you covered a lot, so just to expand just a little bit from from a target perspective, the field of immune checkpoint just keeps increasing. I think the numbers, if you just go on PubMed, probably keeps increasing. It was 40, 50, 60, maybe more, and some are being identified regularly. So it's going to be about finding the one that actually work together the best in some specific patients for some specific type of cancer. For the molecules, you're right. You're looking at basically monoclonal antibodies that can be mono or bispecific, so a combination of maybe 2, 3 antibodies. It's kind of something that that would fit again specific patient and some type of cancer is a big part of the efforts being done today. And then there's another angle to all of this, which is all touch upon that, which is the transcriptional response. And at the end of the day, cancers are kind of our transcriptional response. It is just a manipulation of gene expression, that whether the mutation that leads to that or it's an external factor that makes a cancer cell what it is. So there is technologies now that are being developed to target this aspect of the tumor. And by controlling transcription, you can have a very broad impact because a single transcription factor really controls a high number of targets at the same time. So that's another angle that's being tried as opposed to just on top of what's being done at the membrane level.
Okay, we have a question from the audience. Hello, could you give us an idea of the percentage of cancers treated by immunotherapy? And another question, are there any specificities for clinical trials in immunotherapy? Thank you. Who wants to take it?
Maybe just the first one, because I never run trials with immunotherapy myself. But just the first one, it's about, right now, as I said, there's probably 15 to 20 indications that are covered by immunotherapies. That goes from your lung cancer to liver cancer, breast cancer. That's, again, a list of 15 to 20. Anthony, right there. Now, the number of patients, as I said, is probably only 40% of the total cancer patient population. So that gives you a range that there's still a lot of room to cover to increase eligibility for the patients.
Yeah, and I can cover a little bit the specificities for clinical trials. So right now, in terms of FDA, approved by the FDA and also in Europe, the two main targets are CD19, which is expressed on B cells. And so basically all B cell derived malignancy, lymphomas, chronic lymphocytic leukemia, acute lymphocytic leukemia, all of these are B cell derived. And there's really very nice responses. And also, as Julian touched upon, a lot of toxicity. So this is not at this time type of therapies that are used in first line. They are more used in second, third, or really, when essentially there's nothing else you can do for the patient. And so that's for CD19. Also for myelomas, BCMA, which is B cell maturation antigen, is also expressed on myeloma cells, and that also shows really nice responses. One of the things specific for these is that, yes, you can have up to 80 to 90% response depending on the type of tumor. So it really will melt the tumor away, which is taking care of these patients is quite remarkable. Somebody can come with riddled bones, you do a PET scan and they completely light up, and then, a month later, their PET scan is clean. The disadvantage of this type of thing, not so much for CD19, but for BCMA, it really seems that the responses are not long-lived, where essentially people will relapse with their cancers, maybe 11 months, a median of 11 months down the line. And so a lot of effort is going into trying to figure out what can be done to be able to increase these responses. In terms of T cell receptor therapy, nothing is FDA approved at this point, so it's all in clinical trials. And I would say the furthest along are probably cancer testis antigens, MAGE-A4 and NY-ESO, which is New York antigen. It's a cancer testis antigen, and especially expressed, for example, in sarcomas. But a lot of things are coming down the pike, and I would say a lot of companies are really interested in T cell receptors. both targeting, for example, mutations, driver mutations, P53, KRAS, and also viruses that cause cancer, typically HPV, and which causes head and neck cancer, and also, Merkel cell polyomavirus, but that's really sort of like a boutique, I would say, target. And then also overexpressed self-antigens, such as Wilms tumor antigen 1. So these are not mature yet, but they're definitely coming down the pike.
Thank you very much. It's very clear. Let's say that cancer research has not always been driven by multidisciplinary approaches. For many years, it has been a preserve of cell biologists and geneticists. However, we can see that this field is more and more data-driven and has benefited from several discoveries in fundamental sciences, as well as from crossing between AI and oncology. So can you describe how now it is possible to cross disciplines in a fruitful way as far as immunotherapy is concerned? Maybe Julien, you can start. You are muted.
Of course, I'm muted. It's just the thing of the last 2 years. Sorry about that. So it starts by the fact that the data are actually available for both sides of for multiple fields to look at them. If you start by the problem at hand, which is trying to understand how a cell or set of cells work together, that means trying to understand how 20,000, 30,000 genes can work together. So that's basically territory of mathematics, algorithm, AI, and that's basically, now that we have those data from the clinic and from animal data, that kind of high throughput provide, in a throughput fashion, can provide gene expression, DNA mutation kind of data. You can start to apply, mathematics and AI to this kind of data. So I'd say that it's becoming more and more multidisciplinary just because you can now. And I think that there's also a realization that we have the chance for the first time to move away from just looking at a disease as the product of a single target. Kind of, in the early 2000s, the scientists that mapped the genome, and that was great, but that was a map, right? And now you start to understand how the map works. And I think that starts to trigger a lot of interest, and whether you work just on mathematics, AI, network science, or just biology, where we're intrigued by this and trying to understand how this works. So I think there's mutual interest and the data are available. That's kind of why it's coming more and more multidisciplinary.
Yeah, so I can add on that a little bit. Since also 20 years that I'm in the field. I mean, we started out by doing these little boutique little trials or these small experiments in the lab. But now things it's too much to understand it all. It's just too much for even just one team to be able to understand it. And definitely there's been I would say a quantum leap in the technology used to understand what's going on. And the reason I'm saying that is, for example, once you put, let's say, a checkpoint or a CAR T cell or a T cell receptor into a patient, you can look at the outcome and you can say respond, didn't respond. Okay, but that's really sort of the crudest way to look at to look at things. What we want to understand is, first of all, are T cells persisting, or, for example, the checkpoints, are they eliciting tumor-specific responses? And so we have now sort of the number of technologies that are being used to start to answer this type of questions, not only, for example, flow cytometry, which now has gone all the way up to 40 to 50 colors, but also I would say single cell RNA sequencing. That I would say is really huge in understanding in sort of in an unbiased way what is going on in these patients. For example, I can just give an example. we treated a patient with Wilms tumor and one antigen specific cell. This was a leukemia AML, so acute myeloid leukemia. And we gave him this. So this was a very relapsed patient, not a lot of options. And we treated with the T cells for and in one infusion. And this person had incredible persistence of his T cells. But eventually his leukemia came back and we put more T cells in and there was just nothing we could do. This patient relapsed and died. And so we were really trying to figure out, is like, well, what happened, right? I mean, we had T cell persistence as an immunologist. If your T cells are there, it suggests that something's happening. They wouldn't stay or stick around if there was no purpose for them to be there. And it turns out that it was only single cell RNA sequencing that gave us the answer. When we compared the T cells at the time of response versus progression, we saw that their T cells were very activated, suggesting that they were controlling the tumor at that time. And then at the time of progression, the T cells were sort of quiescent, like if they didn't see the target anymore. And when we looked at the AML using the same technology, we saw that the AML had downregulated some of the mechanism to be able to cut up the peptide and present it on the surface. Thus understanding that the escape was not a lack of target. It still expressed the WT1 target, but it was a difference in how the AML was now cutting up the protein, the Wilms tumor antigen protein, and presenting it on the surface. And this new mechanism, this proteasome that it had switched to, which was a standard proteasome, now was cutting up the peptide in a way that the T cells just could not see it. And so that is, I will say that more and more you will see these trials where a lot of what we call immune monitoring is going to be performed to understand at the individual level what is going on in these patients. And I will say that is very informative because, for example, for this particular case, we actually found another targeting in another peptide of the same protein that could be targeted with another T cell receptor. And so, for example, moving forward, we would use this new target, this new peptide HLA combination, and not the previous one, because we know that there could have been an escape. So immune monitoring definitely will shape what we do next in the clinic.
Thank you. And how, I'm thinking, how is it possible to make different kinds of profiles of researchers working together on this common issue, like a computer scientist, a statistician, a biologist, and geneticists? Is it necessary to build some transdisciplinary kind of nexus-style institute, or is it not necessary? Does every department has its own pool of staff who are heavily specialized in mathematics or in computer science or data analysis, big data analysis? What's the best way to do this?
Okay, I can take a crack at this question. So for me, honestly, all my collaborations come from conferences in person and discussing around a glass of wine, to be very honest with you. And I think that's because science is exciting and fun, and so where people can show what they're working on, and I've been listening to, for example, talks. I was like, wow, that is really something that would really help me for my research. And people are very open to just strike a collaboration and explore different avenues. I mean, that's what, at least in academia, that's definitely, and also in industry, obviously, that's why we're there. But I do miss the in-person because it's much easier. The contact is just much easier.
Yeah, I think we'll all do. Julian, what's your point of view?
So, of course, having 3-4 people in a room, that's the source of innovation. So that always comes down to that at the end of the day. But from my standpoint, so kind of 3 types of collaborations, right? Depending on what you're doing. So academia to academia, academia to private, and private, and those are basically tools available legally to just do this kind of thing. So it's pretty well established, like material transfer agreement. If you want one lab to work with your drug that you develop at the company, all these things kind of exist. So for me, well, from smaller biotech company, it's all about finding someone that has an interest in what you're doing and that can bring something to your field and to the drug you develop or the platform you develop. So common interest, and at the end, it's always driven on our side by just the need, right? When we need an expertise, we'll go find it wherever it is if it exists. So that's really what drives it. Finding somebody who do what we can, where we need them to do.
Thank you very much. So nothing super organized. Nothing can replace the in-person contacts. Maybe we have time for a question in the chat. Yeah, question from Alice. In order to work to help the research in immunotherapy, what kind of studies should I do? So if I were a student, who wants to answer this question? Aude?
Yeah, so I think I'm assuming I'm assuming Alice is a student, and I think follow your interest. I think immunology, immunotherapy is definitely it's coming of age, and there's a lot of labs that are working on it. And if you're, for example, trying to find a lab to work in, shop around, go see what people are doing, what is their research interest, and how you can integrate that lab, either, for example, technician, PhD student, postdoc, and really, I mean, I always counsel to the students, just really talk to as many people as you can. Try to understand what they're doing in their lab and what your project, what the project that you would be working on, for example, depending on your skills. And then also, I think it's important to have a good contact with your mentor. Is that somebody that you actually like to talk to, and that you can work with for a long period of time through, sometimes troubled waters, right? Because science is never, you're not going to, find something immediately. It's usually just a lot of work and a lot of grinding and searching. But really, I always say, look at what really excites you and be led by that.
No, I mean, I completely agree. Talking to people, that's the key. Talking. That's you understand more. Finding a mentor. If you don't have a mentor, it's just hard. It's not just in science. It's just the people open doors for you when they're your mentors. So the advice that Aude said, I completely agree. Talk to people. Pick up the phone. Don't hesitate. Pick up the phone. People actually answer, and people are actually happy to answer and to help. So and never be intimidated. Just somebody who even has a Nobel Prize and you want to work in the lab, call them up.
Okay, so what I understand is immunotherapy, since it's a more and more multidisciplinary topic, there is different ways to tackle this issue, and you don't have to make one kind of studies in particular. Okay, super interesting. Now, so we tackled already the problems of crossing disciplines. Let's talk about the issue of crossing borders, because obviously, depending on disciplines, internal collaborations are more or less suited and can provide different things. So, in your opinion, how can international collaborations foster collaboration? What do you think about it, Aude?
Yeah. So, as we talked, now I would say most conferences are really international. And so that's definitely a great place to start looking at what other people are doing and the collaborations. The research done in Europe is usually, I would say, complementary to what is being done in the United States. I think both continents have their advantages, and so I think trying to foster that as much as possible, like you guys are doing, is definitely, and the story about the Pasteur and the COVID. I think that is really a typical example of across the border collaborations. And I don't know. One of the things I don't know so much is clinical collaborations. I would love to see more and more clinical trials being open in Europe and in the United States at the same time. I think that would be definitely like a plus. Obviously, there's regulatory barriers and financial barriers, but that's something I think we would all benefit a lot from, this type of across the border work.
But when you say research in Europe is complementary to the research in US, could you develop a bit this point?
Yeah, I'd say that. I mean, from my experience, what I've seen is that there's a lot of in the United States, it's sort of, it's very it's immunotherapy or biochemistry. I mean, I would say it's a little bit per track, right? Because a lot of the researchers are also going to be funded, for example, by the NIH, which has its set of priorities. And in Europe, the set of priorities is a little bit different. And so I find that there's a lot of alternatives that we don't do in the United States. For example, we're collaborating in a lab which specializes in the proteasome, and there is no lab that specializes in the proteasome here in the United States. And so that, for example, is so looking, and that's really, I would say, giving weight to the fact that multidisciplinary, go to conferences, understand what different institutes are doing, is definitely a plus.
Okay, Julien, could you what's your opinion?
So my expertise, or at least my experience, has always been at the preclinical level, collaborating with labs in Europe. I've never done clinical trial in Europe, just in the US. So I can really speak to this. And as I was saying earlier, it's driven by the need. You find a lab that actually does a animal model that nobody has. So you just call them up and see if they're willing to work with you, right? Or you work with people like you, and the work you're doing, we can clearly create collaboration because put people in front of you that you didn't think of. So I think both, one, the need, they have a clear need, and you try to identify on your own people who just do that in Europe or somewhere else, or people like you just bring them to us. So it goes both ways.
And what about data sharing? Because I mean, it's pretty difficult to transfer or share data, because I guess medical data is very sensitive, right? It's most of the time it's a very personal, in the sense that you can be identified. You can identify a particular individual with this data. What about the problem of data sharing? Is this predicament to the international collaboration? Because if a chercheur in EU has data and a chercheur in US has data, it's not so ideal to share it. So do you have any thought about it? Did you even consider federated learning or stuff like that?
Yeah, so I really think there's a way around that. I mean, we do it. For example, we do clinical trials in a academic institution, and these are sponsored by companies, for example. And so we run into exactly the same issue. And so all of the identities of the patients are de-identified, but we keep the fundamental data, and so that's definitely I don't think that's a barrier, honestly. What is a barrier is, I would say, you need huge servers now to be able to store all the data that you have accumulated on your databases, like whole exome sequencing or single cell RNA sequencing. This is huge data files. And so to find ways in the cloud to be able to share that. But I think that's a barrier that's slowly being brought down, I will say.
Yeah, and I agree. So yeah, this is regulated, as I said. You can't just do whatever you want. There is a process depending on the type of they have to go through, depending on the type of data you want to share. And in fact, the FDA pushes you to just show data and publish data. Like now, clinical trials results have to be published on clinicaltrials.gov. You have a period of time to do that, but you have to publish them. So the efforts are more toward transparency as opposed to the other way. And when it comes to individuals, it's regulated, so you cannot share data typically. I would say in general that are associated to a name. It's been they identified as I would say. So that's process for that.
Yeah. So and actually, you brought the clinicaltrials.gov, and so I think it's not only U.S. trials that are on there. I think it's also international trials, and I think that's a fantastic way. For example, if a patient has a rare disease or is looking for a trial, as to go on that website and just figure out whether there is a specific clinical study for them, and we have been. We have been contacted by international people, for example, for some of the trials that we did. So it's not so much about finding the trial for these patients, but then it's really the funding. Meaning, for example, the insurance company from the international country will not reimburse the cost for a trial in the United States. So that has been, for example, for sharing patients, that has been a barrier. And that's my point is it would be fantastic if we could open these trials in Europe and the United States at the same time.
Great. Let's take a question from the viewers. So we have here a question from AMHS. From an industry perspective, what do you think would need to happen or loosen up in academia in order to foster better collaborations between companies and labs? Julien, maybe you can start?
Sure. So it's pretty well established in the US. If you take the Silicon Valley or like the Boston area, and it's not the only one, San Diego is the same, there's a critical mass of researchers, universities, and critical mass of entrepreneurs and financiers to make those things happen. So there's this collaboration happen, and I would say bring innovation from academia into the industry is well oiled. Stanford, for example, is particularly good at that. They actually encourage their PIs to basically create companies. So you'll find tons of entrepreneurs within Stanford, the same with Berkeley. So I think that we don't really have a lot of barriers. Typically, as a company, when you approach a university, you have very little leverage to negotiate, like a licensing agreement. You just have to live with it because they're bigger than you. And if they don't want to license technology, they just don't. So it goes, it's a little bit more in their favor. But there is ways to basically just, license technologies and work with scientists with those academic centers. In France, I guess the way I look at it, or in Europe, is much more kind of a mentality or an indication about the process, because the critical mass is not as big. I think it's growing now, and it's going to get there. But it's more, this fostering the entrepreneur spirit from academia is probably what needs to be boosted probably in France to get to where the US is.
Yeah, so and I'm going to just add to that. So, for example, all the clinical trials that we do in academia, they're basically, because it's T cells, it's a privatized medicine, essentially. There's no way we can pay for them if we don't have a corporate sponsor. And so so every single trial that we have, even if it's also funded by the NIH, has a corporate sponsor. And so our business development team at the Hutch, for example, and this is across institutions in the United States, are seeking for investors for the research that we do in-house. And that's how it happens in most academic centers.
Thank you very much. Then maybe we'll take one last question from the public. A question from Eloise. Immunotherapy is often very expensive and not everyone can afford it. How can immunotherapy be made more accessible? How soon can we expect to democratize this solution? Aude, maybe you can start.
Yes, I think, this is an excellent question. And I absolutely agree that right now immunotherapy is too expensive. I think, for example, to undergo a CAR T-cell therapy, I think the minimum, just the product, okay, not even the hospitalization, is $500,000 or $450,000, somewhere around those lines. So I think, what you have to sort of look is a little bit the big picture. These are the first therapies that come and shows that it works. And I believe there's a lot of research also, to try to make this type of therapy much more accessible, also in poorer countries, or to be able to treat more patients. And so one of the efforts, I will say, is there's a bunch of efforts and companies looking into trying to, right now, in order to make a T-cell product, you have to have a huge BSL, you have to have a cell processing facility, which you look for, it's very regulated, if you will. But a lot of companies are looking into trying to make this in a box. And so imagine that instead of having a huge facility with a lot of people, you can actually automatize and put things in a box. And I think more and more we're going to see this type of therapy being developed as immunotherapy. It's going to take a while, though. It's not going to be immediate.
Yeah, do you have a?
So I agree. So it's obviously a very complex. It's not a binary answer we can provide here, but it's driven by the cost of developing drugs. It's developing a drug is a risky endeavor. It's going to be very expensive. It's going to be very long. So the company is not just the cost of goods, as Aude says. Maybe it costs $500K for a course of therapy, but it costs hundreds of millions to get there, and your companies need to get their money back to be able to continue to innovate. So probably the solution is going to be, as Aude says, based on technology. The better we'll be at predicting the outcome of a clinical trial, the better we'll be at anticipating better, developing better therapeutic strategies. Or when I say better, it's having a higher success rate than we do today. All of this will basically trend overall to reduce clinical trial time, clinical trial costs, and that's the only way probably regulation will allow you to just not have this kind of expensive, long clinical trials. It's just for us to get better would be probably the solution long term.
Thank you very much. So to sum up, we can say that immunotherapy is a very promising set of approaches, that it's more and more multidisciplinary. It will require more and more collaborations, but there are still a lot of steps to get there. So thank you very much, Aude. Thank you very much, Julian. And thanks to all the viewers and for their very interesting questions. And let's meet tomorrow for another panel. Thank you very much.
Thanks, everyone. Bye.